Art Davis is the director of the Stanford Center for Human Systems Immunology and the Avery Family Professor of Immunology. He received his bachelor's from Johns Hopkins University and then a PhD from the California Institute of Technology with Leroy Hood. He later was a postdoctoral and staff fellow with Bill Paul, who we heard about earlier today at the Laboratory of Immunology at NIH before joining the Stanford faculty. He's extremely well known for identifying many of the T cell receptor genes which govern T cell specificity, and his work characterizing their binding properties and behavior on cell surfaces, including demonstrating that T cells can detect and respond to even a single antigenic peptide MHC complex. He and his colleagues also developed a novel way of labeling specific T lymphocytes, peptide MHC tetramers, which is now widely used in both clinical and basic immunology studies. His current research focuses on human immunology, specifically a systems-level understanding of the immune response to cancer, vaccination, infection, and autoimmunity, as well as developing newer tools to better understand human T cells responses. While it's very likely that human immunity will develop novel mechanisms compared to inbred mice, there has not been a good system to carry out mechanistic studies in an entirely human system. Immune organoids and combinations with other tissues may be the answer to this fundamental problem. Professor Davis is the recipient of many honors and awards, including memberships in the U.S. National Academy of Science, the National Institute of Medicine, and the Royal Society. So rather than go into more accolades and about Dr. Davis, I'm going to hand right off to you and invite you to give your presentation, please.
So we'll get right going here. Obviously, human immunology is critically important for understanding a lot of diseases. Many diseases impact the immune system. Probably a lot of diseases we don't even know are involved in the immune system are likely to be certainly all infectious diseases, all classical and some non-classical autoimmunities are as well. So it's a major life support system that is so complex that we definitely have a lot of trouble understanding it. And so the inbred mouse model has been terrific in terms of simplifying a basic mammalian immune system, and it's been an engine of modern immunology. But we're seeing more and more that it's not the whole deal. Most mouse models of human diseases have not yielded clinically actionable intelligence. Many vaccines, some for malaria and TB and HIV, work brilliantly in mice and then fail in very expensive human cohort studies. And then we don't even know why they fail because we don't really have enough information on what's going on in people's immune systems. There are differences in immune architecture. There's definitely, of course, a lack of genetic heterogeneity on purpose. It made them inbred because it makes life simpler, but it's not the human reality by any means. We also tend to keep them in very isolated environments, away from any kind of disease, wrapped in HEPA filters as a standard of care. So it's nothing like us in terms of our constant bathing in microbial things. And there also are unique genes and expression patterns, which we're still in the process of finding some of those things out. But I also think that one of the what could be the major reason for why I'm optimistic that we'll find new mechanisms in human immune responses is that there's a huge difference in longevity and fecundity. That is, if a mouse lives for 6 months, that's fine, you're going to have a lot of mice. But if you can't get a fair number of human children to survive for 20 or 30 years, you're done as a species. So there's something like a 50-fold greater window of vulnerability. And we actually even know that human children are typically uniquely vulnerable to infectious diseases up until the age of 5. when the mortality curve gets better. But it's really before vaccines, it's a very steep curve. And that was, reason why vaccines have worked so well is that it's basically eliminated much, although not all, of child mortality. So all this leads me to think that we need to get into human immune systems in a big way, more than we have, and that we're going to find things. And so I think for scientists, pioneering territory, and also not inconsequentially, we should do much better in terms of translation, in terms of understanding diseases and finding ways to cure them. This is just some of the vaccine failures. It's a thing that's gone on for quite some time. Even the SARS-CoV-2 have been relatively successful. There's just been continual failure in some of the major diseases of the third world. And we don't know why in most cases. So this led us to actually invent a number of new techniques and methods. But the one that I wanted to tell you the most about is this, which is immune organoids, where basically we found that just by chopping up tonsils, which are taken out for various reasons, mostly in the West for sleep apnea, Lisa Wagner was able to show that just making a single cell suspension of tonsils and then putting them in a dish, basically they created through self-association a lymph node-like structure such that we could expose them to vaccines, and then they form germinal centers and dark zones and light zones, and make antibodies after a week or 10 days, high affinity antibodies for flu in this case, and also affinity maturation. If you delete the high affinity B cells for flu, in this case, this was staining here, so that you get nothing left. But then you incubate those cells with the flu vaccine, you will get back high affinity B cells. So that suggests that not only are there germinal centers sitting there looking nice in the dish, but they're actually functional in terms of selecting for high affinity. And we've also shown this at the single cell sequence level. And so one of the long-standing problems we've turned some attention to is the problem of influenza strain bias, that we really, for more than 70 years since the introduction of multi-strain flu vaccines, people have noticed that most individuals, like 70%, have a bias towards a particular strain. And that's what they make the most antibody response to. And they sort of neglect the other ones. It's not an equal, even you have 4 strains in the vaccine, you don't get 4 equal responses. And so the dominant idea for 60 years in this business has been called the doctrine of original antigenic sin, which is kind of a very freighted term, but we won't go into that, from Tom Francis. And this idea was that the first flu infection you ever get biases towards that strain, and that's what you're stuck with. And this is sort of what it looks like at the tonsil organoid level. This is a standard flu vaccine called IIV. And this is a particular tonsil that actually this person likes the H3 strain. And so you look at the antibody response to the vaccine, and you get mostly H3 specific antibodies, and not much to write home about in the other strains. So that's the strain bias problem. But is it really original sin, so to speak, or not? And so this has been the work of a very talented postdoc, Vamsi Malajosala, who basically challenged this notion. And I don't have time to show you, but using twin pairs, which monozygotic twin pairs share bias, but they don't share cross-reactivities, which is not consistent with the original sin model. And also with Mary Stott, we're able to look at kids that were unexposed to flu because they're monitored every week, and yet they still had a strain bias when exposed to a vaccine. So this really says this original sin hypothesis is not really operative here. But the twin study says there's a strong genetic tendency. But what is that? You know, that can be all over the place, or it could be something specific. And this is the second bright idea that Vamsi had, which is looking at the way in which B cells get T cell help. They're basically pulling in specific antigens with their specific antibodies or BCRs on the surface. They pull those antigens in, they chop them into peptides. Those peptides then bind to class 2 MHC molecules. And if the right T cell comes along that recognizes that peptide MHC complex, that T cell then provides help in the form of cytokines to the B cell. This enables this B cell to mature and secrete antibodies and class switch and other good things. So and since we know that class 2 MHC is very polymorphic, he guessed that this could be the real choke point. And in fact, in an analysis using the immune epitope database, where you could basically plug in a protein sequence and ask how many peptides will be made by a given MHC molecule, he could clearly show that, for instance, these class 2 MHCs really like H3 peptides, H3 molecules, make lots of peptides. They really hate the B strain, and they're sort of lukewarm with H1. And yet, there are other class 2 molecules that really like the B strain and hate the H3 strain and are sort of in the middle about H1. So anyway, so this points to a possible connection between a strain bias and class 2 MHC polymorphism. Now, is this true? So this is the next brilliant thing Vamsi did, which is he made constructs to simulate the tonsil organoids. And if you just make constructs that are similar to the vaccine, that is, you have separate hemagglutinin strain molecules on different vectors, that is a cocktail of distinct types. These distinct types will then only bind to B cells that have a specificity for this type. There's some cross-reactivity, but mostly it's the type specificity that's important because they're very immunogenic. And so these will all go to the ones that the B cells they like. Now, some will be popular. Say, for instance, the H3 actually is the most popular one. Maybe there are a lot of B cells that are specific for H3. And then, but maybe then there are other ones that really hate the B strain, as we showed. So these will be out of luck. There will be not many B cells and probably not so much T cell help for those. And we also know from other data that T cell help is often limiting for antibody responses. But so this vaccine construct now that combines the different hemagglutinins from flu will go to all comers. All the B cells with any of these specificities will be able to bind and take in this molecule and make peptides from all of them, and then get T cell help from all of them. So that's the logic here, is that this could get a broad amount of T cell help and then maybe equalize the antibody responses. And that's exactly what happens. This is the slide I showed you earlier. Here is the cross-link. Here's where we combine everything. And now you can see you really are getting nearly equal antibody responses to all four strains, because you're providing the missing T cell help for those not very popular B cell specificities. And now to just push this a little further, we're faced with someday we're going to have a bird flu epidemic, and is bird flu very similar to circulating flu strains? The answer is no. Here's a background, and here is a bird flu hemagglutinin. So almost no antibody response. But now if you make a construct that targets bird flu B cells, but you provide T cell help for these the more popular strains, now we can get a huge boost in the antibody response. So anyway, so this is important not just for flu responses, which are it's kind of a not as big a public health problem in some places as others, but um, it's a paradigm for how important T cell help is in antibody responses. And so borrowing T cell help uh, for flu responses or vaccines that aren't getting much antibody response could be a key factor in boosting up and making vaccines successful. Um, so that's one story that we've developed through the organoid, and it came out in Science in December of last year. The other story that has really taken off from the organoid work has been we've known a lot about T cell regulation in the form of CD4 Tregs for a long time. But more recently, it's clear there's another class of regulatory T cells that have a CD8 phenotype. And these are originally described by Harvey Kanter, but we have also now have a bunch of papers in this area documenting that these are yet another important checkpoint for autoimmunity in humans and in mice also. And Mustafa Ghanazada and Xin Chen in the lab basically worked out a CRISPR-Cas9 system for deleting genes so that we could disable these regulatory T cells. And this involves both a FOXP3 deletion in the tonsil T cells and also granzyme B, which is FOXP3 is important for CD4 Tregs. Granzyme B is important for the function of CD8 Tregs. And the long story short is that if you delete FOXP3 in the T cells in the tonsil organoids, whereas normally they wouldn't respond to autoantigens, now you get actually an autoantigen response, in this case to proteinase 3 or double-stranded DNA or histone or snRNP. So it's really dramatic that you're releasing the gates, basically, that the T cells here, the regulatory cells, are suppressing autoantibody responses. The B cells are there, but they don't proliferate and they don't progress to make antibodies. But you delete FOXP3 and now suddenly the gates are open. Also note, the red dots are female tonsils and the black dots are male tonsils. So there's also clearly a female bias, just as there is in autoimmunity. On the other extreme, we see that granzyme B has only a kind of a minor no effect on antibodies. But if you look at autoreactive CD8 T cells or autoreactive CD4 T cells, now you see that either deleting FOXP3 for CD8s or granzyme B for the both CD8s and CD4s, and particularly CD4 cells, which we know are critical for autoimmunity, seem solely to be the province of the CD8 regulatory cells. So this speaks to several things. One is that we're able to do mechanistic experiments in these human organoids. Typically, we've had to go to mice to do mechanistic experiments. And that's a huge limitation, especially if you think they're going to be human-specific immune mechanisms. It's just basically not going to work in mice. So this is like a proof of principle that that this can actually work in tonsil organoids. And this just came out in Nature Immunology, so you can take a closer look at that. We also found a really unexpected thing, which is that deleting FOXP3 actually increased the affinity of antibodies in many tonsils, and compared to wild type. So that says that the FOXP3 cells are not only gatekeepers for autoreactive B cells and autoreactive CD8 cells, but they're also putting a lid on antibody affinity. If you wonder why most antibodies are like 1 nanomolar affinity, it looks like this cell is responsible. So we've also looked at combinations of organoids, and this is work of Elsa Sola and Chen Wang in the lab. And basically what they did is very simple, is just put a piece of skin from an unrelated donor in with a tonsil culture from someone else. And what happens is the tonsil attacks the skin. It's an it basically, it's a skin graft rejection, which is kind of really old time immunology. But it's very clear is skin plus tonsil, you see death, you see lots of T and B cell infiltration, and then cell death in this tunnel assay here. And we can inhibit that with cyclosporin and FK506 and other stuff that's used to prevent organ rejection. So this says that just putting the organoids or tissues together in one dish, you can get some physiologically meaningful immune reactions. So that set us on the path to look for histocompatible combinations. And this has been a great collaboration with Donor Network West, which is the chief supplier of both obtainer and supplier of organs for transplant, the heart, lung, kidney, et cetera. But it also turns out they collect spleens along with everything else, but then they throw the spleens away because nobody wants spleens for transplant. It's not would not be a good idea. So we get the spleens, and we've been accumulating them, and we characterize them. And it turns out they're much more diverse than tonsils, and they support more things more robustly. They give better flu antibody responses. They give yellow fever vaccine responses, and that we hadn't seen before in tonsils. And so we've been collecting them, and they're diverse in terms of the local population. And I think they're going to be an invaluable resource to understand human immune responses at the single cell, at a mechanistic level. And of course, spleens are huge, so you can get 50, 60 aliquots of 100 million cells. So it's a great resource that you can go back to, make organoids and do knockouts and things like that. But we're, and this is some of the diversity of the spleens. I think they're also going to give us a window into human immune diversity, which of course is a huge thing that you can't get a handle on in mice. You've got to have humans, and in this way we have humans better than a cohort. It's because you can just dig into these things with all sorts of molecular techniques that you can't do with living humans. You see variation, and those variations might be very meaningful in terms of why some people respond to this drug or that drug or this vaccine or that vaccine. So I think this is going to be a reason we're creating basically a big spleen bank here, is that we think this is going to be a key for understanding human immune diversity. But our main target right this minute is, can we model things like flu or TB infection in a dish with these organoids? And so this has been collaboration with Calvin Kuo, who's a real expert in organoids on Stanford for many years. And he developed a lung organoid model that works very well. It can infect with SARS-CoV-2, and you can see T cell responses and stuff. And so what we have basically are a system where we're getting lung pieces and spleen from the same donors, and then making organoids from both. It's a different method for lung versus spleen, but then ultimately you can grow them up and then put them together, and then azomosin, the graduate student doing this, infects the lung organoids with a pathogen, either flu or TB. And you look for responses. And so the preliminary results are really looking good, that we see antibody responses. If you infect the lung with live flu virus, we get antibody responses from the spleen. And also T cell responses. We can see that CD8 T cells specific for flu are migrating from the spleen to the lung to presumably to kill flu-infected cells. So we think this could be a great system for testing vaccines in a purely human context. And so that's where we're going with this. We have do I have a few more minutes?
Absolutely. This is fascinating. Please.
Great, thank you. Because this is some really important new unpublished data on TB. And this has been a long-term collaboration we've had with Tom Scriba at Cape Town. And this is a paper that came out just over a year ago, where we're basically looking at a population. So there's this huge adolescent cohort study in Cape Town that looked at 6,000 kids from local high schools, and 44 of them progressed to active TB disease during the study. And so we're able to look at progressors versus controllers. People don't progress. And it turns out that most of their T cell specificities are shared. Almost 99% we see the same T cell clusters. We use this program called Glyph2 that sort of groups similar T cell receptor sequences that we've shown correlates very well with a shared peptide MHC specificity. So we don't know what the specificities are in most cases, but we can track those T cells as sort of biomarkers. And so what you see here is that there's tremendous amount of sharing and suggesting that a lot of these T cell specificities don't matter. But then we look over at the controllers or non-progressors, and we see that there are quite a few dots here that are enriched compared to the progressors. And then similarly, on the other side of the coin, we see a lot of dots that are highly enriched in progressors and not in the controllers. And I've called these distractopes because clearly the one way that a pathogen can get around the immune system is by throwing up a lot of dust, throwing up a lot of specificities. And TB is great for that because it has a huge genome of 4,000 plus genes. So so that was interesting. And then, but we've also more recently collaborated with Change Sedri, who has been working with a cohort in Uganda now that is that are people that are exposed repeatedly to household TB infected people, but they never become quantiferon positive. They're quantiferon negative, apparently for their life, and he has called them resistors. And so we did a study. We made T cell receptor sequences, or a batch of lots of T cell receptor sequences from family members in Uganda that were quantiferon positive, which has been the benchmark for whether you're infected with TB or not, and also T cell receptors with the quantiferon negative people. And then we looked for differences using our Glyph technology. We looked for differences and we found them. We found 24 TCR clusters that seem to be unique to the resistors versus the controllers, or sorry, the quantiferon positive folks. And so, so the question was, can we isolate some proteins for antigens for some of those? And we could, and we found two different peptides, one for eSAT6, which is sort of a canonical antigen that's included in the quantiferon assay, but the quantiferon assay is only measuring gamma interferon production, not anything else. And then the other, and so, but it's very specific for MTB. So it's, if you're positive for eSAT6, you definitely have exposure to MTB. And then we had this other hit, which from a protein called RV2140C, which is a highly conserved protein, and in all the mycobacteria species, including BCG. And in fact, the peptide that we identified in the MHC that it's restricted to, the R11, is identical in sequence to the BCG version. So, so do we see anything? So we're seeing this in resistors, but the question we posed is, well, there's a lot of quantiferon negative people in the Cape Town study. Maybe some of those are resistors, and it turns out they probably all are. That we have looked at them with T cell reagents using these peptide MHC complexes and a multimer format that we made called spheromers. what was interesting was in the quantiferon, 5 out of 5 of the quantiferon negative ACS donors, the Cape Town cohort, they were all positive they all had T cells specific for ESAT6, not incredibly abundant compared to quantiferon positive folks, but they were there, and it was clear that even these people were supposed to be negative for TB. They actually had been exposed to TB, but they're on a different program, and we have a paper with Chayton in Nature Immunology last year where we showed that there are T cell responses that look interesting, but they're quite different, although we couldn't get a lot of specificity in that. And then even more surprising was that when we did staining with the RV2140C peptide, we found a massive response in the quantiferon negative donors from this cohort. 5 out of 5 had almost 1% of their CD4 cells specific. 1% of CD4 is like a huge CD4 T cell response. It's like an active infection. We don't know if these people are actively infected, but there's definitely a lot of T cells there. And it's now the opposite of ESAT6 in the quantiferon positive. We can detect some T cells, but they're not very abundant. And then even more interesting is, uniformly in quantiferon positive people, the TB specific T cells are Th1 star. They just, over and over again, you see this. There are a few other cells, but mostly Th1 star. That's the book, and that's also why it makes a gamma interferon. So that's why the quantiferon assay works. But it turns out that most of these T cells for the RV antigen are follicular helper cell phenotype. So completely different phenotype. And this is a phenotype that actually in mouse studies correlates with protection. So we're talking about a very different T cell response. And in quantiferon negative people, which are indistinguishable in other ways from healthy controls. Of course, there could always be some subtlety here, but so this says really most people in TB endemic areas like South Africa have been exposed, have been infected, and they make a response. But some make a response with Th1 star, and others with this completely different response. And also, it turns out that individuals or younger people in South Africa start out about 45% at age 10 are quantiferon positive, but by the adulthood, they're 75 or 80%. So there's clearly a lot of conversion from a quantiferon negative status to quantiferon positive. And so we've been looking, we just are finishing up a study with children vaccinated with BCG. And what we see is that they are, of course, almost by definition, quantiferon negative, but they're making many of them are making a significant RV response, and it's a follicular helper cell response. So that says that vaccine, which is very protective of children, that's what you start with. And then maybe with repeated exposure to mycobacterium, including MTB, you keep an active response of CD4 cells for things like RV. But, and then at some point, I think the MTB infection overwhelms the immune system and changes it into Th1 star. So that's my story, or the various stories. And I think we're really going full speed on this RV antigen as a potential vaccine candidate that could be way more effective than anything so far. And conclusions are, again, just to say that we really, if we expect to dig into human immunology and things that are not evident in mice, we need a good system to do that. And I propose that these human organoids could be that system. And we've already shown a utility in terms of understanding some basic things about flu antigen preferences and about the division of labor between CD4 and CD8 regulatory cells. and of course, you can measure different vaccine responses in spleen. And now combining lung and spleen from the same patients, we're able to at least model an infectious disease response and in the near future test vaccines. And then in the terms of the TB story, I think it's quite amazing that all the quantiferin negative people, which were thought to be uninfected, have in fact been infected, but that they're making a very different and probably more effective response than quantiferin positive individuals. So with that, thanks for your attention. Thanks for having me, Eleanor. And of course, this wouldn't be possible with some very talented postdocs and students. Vamsi Faye, I didn't mention, but she's doing a lot with the TB story. Meng Sun earlier, Azim, and earlier others, Hong Wang, and collaboration with Tom Scriba and his group, and some also great collaborators at Stanford. So I'll stop and answer any questions. Thank you.